The mechanism behind anemia of chronic disease involves three converging problems occurring simultaneously, and understanding all three is essential to grasping why this anemia develops and persists. The first problem is reduced heme synthesis. Heme is the iron-containing portion of hemoglobin, the molecule that physically carries oxygen inside red blood cells. When heme synthesis is impaired, hemoglobin production slows, and the bone marrow cannot generate red cells at the rate the body requires. The second problem is decreased erythropoietin production, abbreviated as EPO. EPO is a hormone produced primarily by the kidneys that acts as the body's primary signal to the bone marrow to ramp up red blood cell production. When EPO levels fall, as occurs in chronic disease states, and most dramatically in kidney disease, the bone marrow receives a weaker stimulus and consequently produces fewer red cells. While renal disease causes the most profound EPO reduction, chronic inflammatory states can also suppress EPO production independently, further compounding the deficit. The third problem is increased hepcidin. Hepcidin is a peptide hormone produced by the liver that regulates iron availability throughout the body. In chronic disease and inflammation, hepcidin levels rise significantly. Hepcidin exerts its effect by binding to a transport protein called ferroportin, which is responsible for moving iron out of storage cells, particularly macrophages and liver cells, and releasing it into circulation. When hepcidin binds ferroportin, it causes the protein to be degraded, effectively sealing the iron exit. Iron then becomes trapped in storage and cannot reach the bone marrow. In summary, anemia of chronic disease, also known as anemia of inflammation, is caused by cytokine-mediated disturbances in iron metabolism, primarily through upregulation of hepcidin, resulting in impaired iron absorption and iron sequestration in macrophages, which decreases iron availability for erythropoiesis and leads to functional iron deficiency despite adequate iron stores. Inflammatory cytokines, such as interleukin-6, tumor necrosis factor alpha, and interferon gamma, also suppress erythropoietin production, inhibit erythroid precursor proliferation and differentiation, and shorten red blood cell lifespan through enhanced erythrophagocytosis by macrophages, further contributing to anemia. Iron sequestration via hepcidin, IL-6, and other cytokines induce hepatic hepcidin synthesis, which degrades ferroportin and blocks iron export from enterocytes and macrophages, causing hypoferremia and hyperferritinemia. Impaired erythropoiesis, pro-inflammatory cytokines suppress bone marrow erythroid progenitors both directly and indirectly, hamper differentiation, and reduce responsiveness to erythropoietin. Functional iron deficiency. Iron is present in reticuloendothelial stores, but unavailable for red cell production due to blocked mobilization.